Good morning. My name is Chiara Cirelli, and I'm here to tell you a little bit about the functions of sleep. First of all, I want to thank Professor Fiorenzo Conti and the Italian Society of Physiology for giving me this opportunity. So first of all, I want to start by defining sleep, and then I'm going to tell you why we think sleep serves some essential function, and then discuss the ideas about what these functions could be. Now, sleep is first and foremost a behavior. You see here two animal species, the cockroach and the jellyfish Cassiopeia, in which sleep has been characterized. And the first step to do so is to see whether some specific criteria are met that are behavioral criteria. And you see them listed here in decreasing order of importance. So the most crucial one is the increased arousal threshold, followed by the presence of a quiescent state, and then the presence of homeostatic regulation, and then specific posture and preferred sleeping location. Now, by far the most important defining feature of sleep is the increased arousal threshold. The fact that when we are asleep, we lose partially the ability to promptly respond to potentially a very dangerous situation, as shown in this painting. The second criterion is quiescence, at least partial quiescence. Now, this is a very common feature, but it is actually not a requirement. Marine mammals, dolphins, and whales do keep moving when they are asleep, and they can do so because they actually sleep with only half of the brain at any given time. These are recordings, classical recordings done by Mukhametov and colleagues in Russia. And they show that indeed these animals can have one side, here the right side of the brain with slow waves, while simultaneously the left side shows an activated wake EEG. And then after a few seconds, the situation switches. So now is the right side that is awake and the left side is asleep. Now, the third criterion, which is very important, is the presence of a homeostatic regulation. There are complicated mechanisms in the brain that allow us to recover at least some of the sleep that was lost. I show you one example in fruit flies. They were sleep deprived by using this apparatus, so through mechanical agitation. The sleep deprivation was of different length, 6 hours at night, or the entire 12 hours dark period, or 24 hours. Flies are diurnal animals like us, so they tend to sleep mainly during the night. And you see here basically that the longer is the sleep deprivation, the greater is then, after sleep deprivation, the amount of sleep that the flies had recovered. Crucially also, if the sleep deprivation, the mechanical agitation was only done during the day, there is basically no sleep rebound, which tells us also that really the sleep rebound here is driven by sleep loss rather than by the stress of the mechanical stimulation. Now, as I mentioned, there are other features. We all have our preferred posture and sleeping location, but we also know that if sleep pressure is very high, we can also sleep in very awkward conditions and locations. Now, why do we think sleep serves some essential function? There are some in the sleep field, although they are quite a minority, that support the ecological hypothesis, according to which sleep is simply a behavioral adaptation that may be useful in some conditions. So for instance, if it's dark, it's cold, just to stay out of trouble and conserve energy, but really doesn't serve any essential function. Another version of this position is the release sleep is a time filler. If you are a predator, when there is nothing better to do, then you can simply sleep. Why do we think overall that these hypotheses have to be rejected? There are several reasons. One is that sleep is universal, meaning it has been described so far in all animal species that have been carefully characterized. You see some examples here, especially some recent additions to this list. For instance, the flatworm, the Hydra vulgaris, the Cassiopeia that I mentioned before, the fruit flies, and the sea snail Aplysia californica. Now, I put universal in quotes because still we have known about sleep only maybe in 230 or 40 species out of the more than 1 million animal species present on Earth. So we still have a very limited view of sleep and also very biased view because most experiments are done in vertebrates and especially in mammals. Now, there have been some claims that some animals, for instance, some frogs, do not sleep. But as we have argued and others have argued in these reviews, actually this evidence is quite weak, is based on a very limited amount of experiments using, for instance, very stressful stimuli to measure arousal threshold. There have been also some claims that, for instance, some flies may not sleep at all and they may be just fine. But this conclusion was based on the analysis of behavior and movements or lack of movements in flies without actually measuring arousal threshold. And this recent study actually showed that flies do move. For instance, they move the proboscis during sleep. And this phase of sleep with this kind of movements is actually a very deep phase of sleep with a very high arousal threshold. So this is just a reminder again that to study sleep, you really need to measure arousal threshold and you cannot equate simply rest with sleep and movement with wake. Now, another evidence is that there are examples that have been characterized, many of them quite recently, of animals that under extreme conditions can reduce their sleep a lot and still maintain some of that sleep. For instance, the frigate birds, when they migrate for days, up to 10 days over the ocean, they never land, but they still keep moving and they have unihemispheric sleep, a little amount of unihemispheric sleep. The sandpipers during the mating season also reduce their sleep very significantly, and yet they still maintain some of that sleep. The fur seals also are a very interesting model. When they are on land, they have bihemispheric sleep, but when they are in water, they switch to unihemispheric sleep. They reduce sleep, but they still maintain some of this unihemispheric sleep. And so the question is, why would they do so if sleep does not serve some essential function? Now, another evidence comes from the presence, alluded before, of a strong homeostatic regulation. So there are complex processes in the brain that allow us to recover some of the sleep that was lost. After sleep deprivation, sleep usually becomes longer and or deeper. For instance, in mammals, there are more slow waves in rebound sleep. And here you see an example of that. This is in a rat. The top panel shows 12 hours of the light period when rats are mainly asleep. You see color-coded behavioral state, wake, non-REM, and REM sleep. And the black line indicates the amount of slow wave activity, which is basically a combined measure of the number of the slow waves of non-REM sleep and their amplitude. And we have known for a long time that the slow wave activity is very high at sleep onset. and then declines exponentially. And in fact, that's why we think slow wave activity is a marker of sleep pressure as well as of sleep intensity. In the lower panel here, you see that the rat was then sleep deprived for 6 hours and then allowed to sleep. And the sleep now is more consolidated. There are fewer, shorter episodes of wake, and the amount of slow wave activity increases very significantly. Again, why would you go through this process of rebound if sleep was just a time filler? Now, what are the ideas that some of the most popular ideas that we have now about the functions of sleep? And I will focus mainly on the brain because this is where we have the most compelling evidence for essential functions of sleep rather than simply rest. On one hand, we all know why we sleep. If we don't, next day we feel miserable and everything is much more difficult to achieve. And indeed, there is very strong evidence that sleep loss affects very broadly cognitive function. So it's not just that after sleep deprivation, there is decreased vigilance and attention, but also the ability to integrate a lot of information, to learn new things, to remember, to speak clearly, are all affected. Our ability to assess risk or appreciate humor are also affected. And even in conditions in which actually subjects perform more or less at baseline level after sleep deprivation, they often report, however, that it took them a much bigger effort to achieve that level of performance. One evidence that we have that indeed is not just that we have impaired cognition with sleep deprivation, but that then is only through sleep and not simply in quiet wake that we can recover from that impairment. Here you see examples of 3 studies using 2 different tasks that I will describe better when we talk about sleep and learning. But suffice to say here that in this experiment, subjects trained in this task very extensively during the day, and so they actually accrued post-training fatigue. So by the end of the day, they actually had a deficit in performance because they were tired. And here is an example of the original study by Sarah Medling with the visual discrimination task. So subjects were trained 4 times, and they got actually worse. So their deficit in performance increased in the course of the day if they had no rest, or even if they were allowed to rest, which means they could close their eyes for 1 hour after the second session here. Instead, when subjects were allowed to nap for a short period, 30 minutes or 60 minutes, then the deficit disappeared. So again, there is something related to the presence of sleep that allows the maintenance or the restoration of cognition. And we are trying to understand what is exactly that sleep provides to maintain cognition. So that is what we don't understand. What is the process that sleep is providing that is so beneficial for the brain? Before I discuss that, I need to remind you that most of what I'm going to tell you is about mammalian sleep, and in the adult brain, 80% or so of sleep is non-REM sleep or slow wave sleep, and 20% is rapid eye movement or REM sleep. In non-REM sleep, when we record these slow waves, this is because the cortical neurons and thalamic neurons fire with a very specific pattern of bistability, because they are up and firing, they're active for half a second or so, and then they stop firing and they are hyperpolarized for the next half a second, and then they go up again and fire, and then down in silence. And because they do so more or less synchronously, that's why we can record the slow waves on the scalp. By contrast, the REM sleep is also called paradoxical sleep, because in fact the neurons are continuously active and the EEG is really wake-like. Now, the other thing that I need to remind you is that these non-REM-REM phases alternate in cycles. In humans, there are 3-4 cycles. Each of them is around 90 minutes long. Rodents, rats, like shown here, alternates much more frequently. So there are many of these cycles in the course of the major sleep phase. So what are the ideas about the sleep functions? A very classical one is that sleep somehow is for brain restoration, especially energy restoration. So we know, of course, that the brain is only accounting for around 2% of the total weight, but it consumes a lot of energy, as shown here. 20% or so of the overall energy goes to sustain brain activity. And there are many calculations, and although the exact percent may differ in the different studies, but overall, all these experiments agree that the bulk of the brain's energy budget goes around 70% or so to sustain synaptic signaling, synaptic transmission, and the propagation of the action potential. So the energy needs of the brain are higher in wake than in non-REM sleep. This is very well established. Here are classical studies measuring glucose consumption in the monkey cortex, finding that overall that consumption is 30% lower in non-REM relative to wake. And here are similar studies measuring oxygen use in humans, again, concluding that in non-REM sleep, the use is 20%, 25% lower than in wake. Now, this is not surprising because I mentioned before, overall brain activity is higher in wake than in the bulk of sleep, that is non-REM sleep. So here is in rats, this is cortical firing rates that are higher in the wake state relative to the non-REM state. And they are in between in the REM state. And again, this is because in non-REM neurons are bistable, so there are these periods in which they're not firing. Because of that also, if you measure glutamate levels, because most neurons in the brain, and especially in cortex and thalamus, are glutamatergic neurons. So if you measure in the extracellular space glutamate levels, for instance, with voltammetry as done here, you see that overall they are higher during the night, which is the wake period in a rat, and lower during the day. And if you look with higher temporal resolutions, here you see that indeed the levels keep going up in wake and down in non-REM sleep, and then up again during the short periods of REM sleep. So again, all this is not surprising. Then obviously neurons are more active in wake, they consume more energy. Sleep overall, because of non-REM sleep, seems to be a condition that could allow, if there is depletion of energy reserves during wake, a restoration of those reserves. Here is one version of this general idea of brain restoration, energy restoration. This hypothesis was proposed by Joel Bennington and Craig Heller. And it was that, again, brain activity in wake brings about an overall depletion of the energy reserve. In the brain, glucose is stored as glycogen, which is stored in astrocytes. So the idea is the brain glycogen gets depleted. This causes transient cellular deficits, the accumulation of adenosine as a byproduct of metabolism. Adenosine then acts to promote sleep, and specifically slow wave sleep, and this is then conducive to the restoration of the reserve of the brain glycogen. Now, this hypothesis has been tested quite a lot, and some aspects of this general idea have been confirmed, specifically related to adenosine. So we now know, and here is the original study by Porca-Iskanen and colleagues, that indeed adenosine levels in the brain, in the tissue, are higher in wake than in sleep, and specifically in slow wave sleep. And if you do sleep deprivation for several hours in a rat, you see that adenosine levels keep going up. It was also shown by many studies that are summarized, for instance, in this review, that adenosine then is able overall to promote sleep because it inhibits many of the arousal systems in the brain. And on the other hand, it is able to excite some cellular groups, especially in the hypothalamus, that are sleep promoting. So overall, this general idea that adenosine accumulates during wake, and this accumulation somehow reflects sleep need and then promotes sleep, has been confirmed. And it's not, of course, by chance that caffeine, which is the most powerful and common wake-promoting drug that we all use, is indeed adenosine antagonist. It has been more difficult, though, to actually test and confirm the other side of the story, meaning that really sleep serves to restore brain glycogen. First of all, in some studies, indeed, the overall amount of glycogen in the old tissue was found to be higher in wake than in sleep, but it was also found that these changes are very rapid, that already after a few minutes of wake, glycogen levels get depleted and then they get restored by sleep. Other studies have found inconsistent results. They have not been able to confirm this across different animal species of different brain regions. A major problem is that it's difficult to measure the tissue levels of glycogen because it gets degraded very quickly. We have been trying to test this idea using electron microscopy and literally measuring the size and the number of the granules of glycogen in the astrocytic peripheral processes close to the synapses, which is where they should really provide the energy that is needed. And we overall found some evidence that indeed the turnover of these granules is increased in wake. That we wake, actually their number increases, but their size decreases, and therefore their ability perhaps to release glucose residues is impaired. But overall, again, this side of the story about glycogen remains quite debated. And more generally speaking, the general idea that sleep is to restore energy reserves on the moment, on the spot, every day, is challenged by the presence of REM sleep in itself, because REM sleep is a metabolically very active state because neurons are tonically firing. And indeed, here in humans, when you measure oxygen levels in wake relative to REM sleep, you see no overall difference. And perhaps even more importantly, REM sleep accounts for 20% of sleep in the adult brain, but is 50% or more early on in development. And it's difficult then to imagine that all this sleep that is so metabolically expensive is actually used to restore energy reserves. Now, the second general idea that I want to discuss is about the role of sleep for waste clearance. And this is in relation to the recent discovery by Mike and Nedergaard and colleague, of what they call the glymphatic or glial lymphatic system. Glial because they found that a major player in this paravascular system is the astrocytes, and specifically the aquaporin 4 water channels that are expressed in the enphytes of the astrocytes. So this system is, as I mentioned, paravascular. So according to the original rendition of this model, the cerebrospinal fluid from the subarachnoid space here continues into this periarterial space that is around the arteries, and you see here a penetrating artery. Then the CSF is exchanged through the facilitatory effect of the water channels with the interstitial fluid in the parenchyma, and mainly through, but not exclusively, but mainly through convection movements, the fluid as well as the solutes in the parenchyma are then moved towards the perivenous space, and through this space then they're moved out of the brain. Now, there is another component of this system, because we know actually that a lot of the waste solutes from the parenchyma seem to be moved out of the brain also, not just along the veins, but also along the nerves here, the cranial nerves. Now, the glymphatic system has received recently a lot of attentions for two reasons. One is because it was found that the activity of this system is much higher during sleep than during wake. And the second, for the long-term implications of this activity in terms of neurodegeneration. So here is the original study linking the glymphatic system with sleep by injecting a tracer in the cisterna magna. The experimenters found that then the ability of the tracer to diffuse around the arteries and then in the parenchyma was much higher if the animal was asleep than if it were awake. They also found that this difference was due to the fact that in sleep, the interstitial space, the extracellular space, is larger, up to 60% larger than in wake. And finally, they also found that they could basically measure a similar difference in the ability of a dye to diffuse into the parenchyma if they have two wake conditions. One is the control, and the other one is wake with low levels of noradrenaline due to the injection of a cocktail of noradrenergic antagonist. So the implication here was that perhaps sleep helps, especially because sleep is associated with low levels of noradrenaline, and presumably noradrenaline acts acts on the astrocytes and perhaps on the astrocytic processes, and this action then leads to these changes in the extracellular space. Now, the link with neurodegeneration was provided already in this original study, because the authors found that if you inject into the tissue, in this case in the frontal cortex of a mouse, the beta amyloid peptide, and then you measure over 4 hours how much of this peptide is retained in the tissue, they found that the clearance was much quicker if the animal had been in sleep or in ketamine xylazine anesthesia rather than awake. And so the implication again is that the clearance of these solutes, including toxic solutes like beta amyloid, is much more efficient in conditions associated with slow waves than in wake. Now, a lot of what we know of this system comes from rodent studies, but recently, this study in humans seems to suggest that perhaps something like that also happens in humans. So here is the dye, the tracer is an MRI contrast agent that was injected for clinical reasons intrathecally. And then here there were two groups of patients. Some were allowed to sleep and others were sleep deprived. And what is measured here is again the ability of the subject to clear this tracer measured in the cerebrospinal fluid, depending on the condition. And you see that eventually, but much later, after weeks, both groups of patients have cleared completely, but for a long period of time, the group that could sleep had much larger clearance than, which is the blue one here, much larger clearance than the sleep-deprived group. And so, again, overall, the implication is that then perhaps if we sleep well, the clearance and all the toxic waste that is being accumulated in wake is removed much more efficiently during sleep. And this may have long-term consequences for neurodegeneration. Now, there are many open questions. One is, again, what is exactly in sleep that is allowing this clearance to be more effective? Is it the slow wave and or the low noradrenaline levels? The implication being that if it is just noradrenaline, of course, REM sleep could also be important because the noradrenergic activity is very low, actually lower in REM sleep than in REM sleep. While if it is just the slow waves, of course, then REM sleep would not be beneficial at all. The other issue is that, again, with the exception basically of the study that I mentioned just now, we know much less about exactly how this glymphatic system works in humans relative to rodents. And the big question is also in terms of exactly what is the advantage that sleep provides in the long term in terms of neurodegeneration. The protection that sleep provides, is it passive or active? The results that I've shown you so far would assume that is an active process because, again, because of this increased extracellular space, really sleep actively promotes the removal of the solutes. The other possibility that is not mutually exclusive, though, is that sleep is simply providing a time in which these beta amyloid and tau are not accumulated. And this is because we know now that indeed this neuronal activity that drives the increase in these compounds, both of them are activity dependent. And this has been shown both in rodents and in humans, in which sleep deprivation of being awake indeed increases the levels of beta amyloid and tau in the interstitial fluid, and in humans it has been shown in the cerebrospinal fluid. And in rodents, there is direct evidence, as shown, for instance, in this study, recent study by Holth and colleague for tau, that the accumulation during sleep deprivation during wake is really depending on the fact that neurons are more active. And here you see the red is the condition in sleep deprivation in which tau increases, but even with sleep deprivation, this does not happen if neuronal activity is blocked by this toxin TTX. So the question is sleep good to avoid overall long-term neurodegeneration simply because it's not wake, and so because the neuronal activity is lower, or is it good because indeed there is active process of clearance, or perhaps both. And again, this has implication for non-REM versus REM sleep, again, because REM sleep is such an active, metabolically active state. I want to discuss now the role of sleep in learning and memory consolidation, as well as more generally the role in synaptic plasticity and synaptic homeostasis. There is a very large body of literature on the beneficial effects of sleep on both non-declarative and declarative memories. And I can only give you a few detailed examples of these studies, and then I want instead to discuss more generally the open questions that are still remaining in this field. So if we talk about non-declarative memories, they include motor and sensory skills. These are memories that are usually acquired quite slowly. They show little generalization, and typically they are encoded explicitly, meaning that the subject makes a conscious effort to learn. Here is an example of a task that has been consistently shown to benefit from sleep. In the rotation adaptation task, the subject is in front of a computer screen and is fixated in the center of the screen, and then is asked to reach with the cursor one of the 8 positions that gets suddenly illuminated. And the instruction is to do this as accurately and as quickly as possible. Now, unknown to the subject, in fact, there is a systematic rotation that is being imposed. It's very little, but is always present, and to the same degree, to the perceived trajectory of the mouse. But the subject doesn't realize that and doesn't see the movement of his or her hand because this is covered as shown here. So this is an implicit form of learning. And it was shown that if you train in the evening and then retest in the next morning, there is an improvement, but this improvement does not occur if the same number of hours are spent awake. It was also shown in the original study by Huber and colleagues that after learning during sleep, there is a local increase in the slow wave activity, specifically in the right parietal cortex, which is a region that was shown previously to be engaged in learning this task. And it was also shown that this local increase in slow wave activity was positively correlated with the improvement in performance in the task the next day. So this suggests that slow waves are important for this task. And this finding was further strengthened by another study that showed that if you do a selective deprivation of slow wave sleep by using a mild acoustic stimulation that does not wake up the subject, but it simply reduces the amount of slow wave. In that case, the benefit of sleep for this task is completely abolished. So it seems that this task is especially improved based on the slow wave sleep. We also know, as I mentioned before, that if you train too much, too extensively within the same day in this task, you actually develop post-training fatigue, and that fatigue can only be recovered by a nap and not by spending the same amount of time in quiet wake. Here is another task, the visual texture discrimination task, that we know has been shown consistently to benefit from sleep. This is quite a difficult task in which the subject has to see this image very briefly for only 10 milliseconds, fixates on the center of the field, and then is asked about the orientation of 3 lines in one of the 4 quadrants. So the training is always done specifically in one quadrant. And then there is a delay of variable length, after which a mask is presented always for 100 milliseconds. Now, by design in this task, the accuracy of the subject is maintained quite high, to 80% of correct responses. And what changes and improves during the task training is simply the speed. And this happens by progressively reducing the delay between the presentation of the stimulus array and the mask. So here is the, on the left, the typical time course in one single subject of the learning in this task. So here there is one training session every day, and you see that most of the improvement in performance, meaning again, the decrease in the latency between the stimulus array and the mask, occurs in the first 4 or 5 sessions, and then performance plateaus. Now, it was already noticed in this original study that if you try to actually do 2 sessions in the same day, there is no obvious improvement in performance unless there are at least 6 hours between sessions. This already suggested that there was some kind of offline consolidation and perhaps sleep was involved. This was tested in a follow-up study by the same authors that showed indeed that every time there was nighttime and sleep between the 2 sessions, for instance, from here to here or from here to here, there was a notable improvement. Instead, in most cases, when the sessions occurred in the same day, like from here to here, there was no improvement. Now, since this original study, there have been many other experiments conducted by different labs across the world. So we know that this is a consistent finding that this task indeed benefits from sleep. We know that it's not just one night, but also a long nap is enough to see a significant improvement in performance. We also know that similar to what I've shown you before, also in this task, the benefit of sleep disappears if there is a selective suppression of slow wave sleep. Again, suggesting that this type of sleep is very important. But there are other studies suggesting that, in fact, is probably the entire cycle. So it's important to have consolidated the sleep in which non-REM is followed by REM sleep for the benefits of sleep in this learning. Also, as I mentioned before, again, if you train too much in this task within the same day, then the fatigue can only be recovered with a nap. Now, so the story about this task is simple. There is a clear-cut benefit, but overall, the story for the role of sleep in non-declarative memories is more complex, because we know of examples of tasks that do not seem to benefit from sleep. And this includes even tasks that are discrimination tasks that at least superficially seem to be quite similar to the visual discrimination task that I just described. For instance, some of these studies have tested the role of sleep in discrimination of faces or in the discrimination of specific shapes like this one and have obtained negative findings. Now, the take-home message is that we still don't understand why this is the case, why there are some tasks that do not seem to benefit from sleep. Is it just the amount of training that is being used? There may be a lot of inter-individual variability. Is it the complexity of the object that is being, of the image that is being shown? Does it matter whether it's shown on the center of the visual field or in the periphery? The bottom line is we don't know. Now, what about declarative memories? These are memories of single events in our life, episodic memories, or more general knowledge, so semantic memories. Generally speaking, these memories are quickly acquired, but they are also quickly forgotten, and they are hippocampus dependent. Now, here again, there is a huge literature, so I will only give you again a few examples and then the open questions that are remaining. Here is the pioneer study that provided the initial suggestive evidence that sleep may be important in slowing down the forgetting that happens always after encoding this kind of declarative memories, unless, of course, you retrain. So this study by Jenkins and Dallenbach in 1924 had only 2 subjects, and they learned a list of nonsense syllables, and then they were tested after 2, 4, 6 hours, and those hours were either spent in sleep because they slept in the lab, or awake because the list was presented in the morning. Now, overall, the result was that in both cases there was forgetting, but the rate, the speed of this forgetting was slower if those hours were spent mainly asleep. Now, this was, of course, a very limited study. There were many limitations. There were only 2 participants. Sleep was actually not recorded during the night, etc. But the fundamental result has been confirmed. Here is just one example. This study was done by Jan Born's lab, includes many different conditions and controls. These are students learning vocabulary words. And as you see, the learning part can happen in the morning, in the evening, and then is followed immediately by sleep. But actually the retesting, the recall, is done 2 days, 2 nights later. Or instead, learning is immediately followed by sleep, and then the recall is the following evening. In some cases, instead, there is learning and then sleep deprivation. So you see there are many, many conditions, and the overall results are shown on the right. First of all, the D panel shows that indeed what counts is whether immediately after learning there was sleep or sleep deprivation. So the forgetting is much higher with sleep deprivation. Similarly, it matters whether the time of learning was in the morning, then there is forgetting, or in the evening. In that case, there is very little forgetting because the subject goes to sleep immediately afterwards. Instead, there is basically no, it doesn't matter whether the recall happens in the morning or in the evening, and also within 24 and 36 hours, the duration of the retention interval does not seem to matter. So this was the first suggestion that indeed it seems that it's very good that you learn and then you go to sleep immediately afterwards to reduce the amount of forgetting. Now, the discussion and the follow-up experiments by several groups has been in terms of trying to explain why sleep is able to slow down the forgetting, which is a difficult question to address because, after all, we know very little about the mechanisms that are accounting for forgetting. So one possibility is that we forget simply because the memory traces that have been laid down due to the original learning are literally destroyed and overridden by new traces, because we are always learning something new. So in this view, of course, then sleep is helpful, but it is so simply because sleep is not wake, and so there is no new encoding during sleep. So this position, of course, ascribes to sleep a kind of a passive role. But others in the field instead are suggesting that sleep may have a much more active role. It can actually help directly in consolidating, in making memories stronger and more resilient to forgetting. And so one way to try to test this second position, as was done in this pioneer study, was to make the interference during wake especially strong. So the design was the following. You learn, in this case, A-B pairing paradigm early on, and then you go to sleep, or you learn in the morning and then you are awake for the entire day. Before being retested here in the A-B training, you are actually asked to train and to learn a new paradigm, which is the AC training, that is designed to create a lot of confusion, a lot of interference with the material previously learned, so the AB. So the idea was that if sleep here has provided an active protection, then despite this large interference challenge, you should see a difference between the sleep condition and the wake condition. And indeed, this is what was found in the original study. So when you do the AB training here, and then you are tested after sleep or after wake with no interference, there is a little advantage of sleep. But the advantage is much larger, again, in this more difficult condition. Now, this, again, was supporting the idea that sleep may have an active role in consolidation. The problem right now in the field is that this original study has not been replicated. There have been 2 attempts by other groups to really perform basically exactly the same experiment, but the results have not been consistent with the original one. So the fact that sleep may have some specifically active role in the consolidation, the evidence for that is still quite mixed. Now, there are other very interesting open questions. Some of those are discussed in this very recent brief review. For instance, there is evidence that sleep benefits some kind of memories, of declarative memories rather than others. For instance, emotional memories rather than neutral memories. But again, also here, there are positive as well as negative findings. We also know that sleep can promote the gist extraction, as well as they promote insight, so the sudden realization of a hidden rule, for instance. But also this has been found in some studies, but not in all of them. And we don't really understand why this is the case. One probably again, a major reason for this variability is that we still don't understand exactly the mechanisms, the synaptic cellular mechanisms by which sleep benefits memories. And this I will discuss at the end of this talk. Now, what about sleep and future learning? So there is very little, if any, learning during sleep, but certainly we all know that it's so much better to learn new stuff after we have had a very good night of sleep rather than a poor night of sleep. So this has been shown in specific experiments for single item learning, motor skill learning, and item context binding learning. So both for procedural, non-declarative, and declarative memories. Now, it would seem that this is quite trivial to show, but in fact is not trivial at all. The reason being that, of course, when you ask subjects, let's say in the morning, to learn new material, and before that they have been either asleep or sleep deprived, if you see a difference and the subjects allowed to sleep are better, it could very well be that the reason is that the sleep-deprived subjects simply did not attend, did not encode, because they didn't even have, they didn't pay attention to the images. So that because simply there is decreased vigilance levels, arousal levels. But these three studies specifically went a long way to correct for that. So to make sure that the level of attention and vigilance is the same. And despite that, they could indeed see that sleep promotes future learning, again, for different kinds of tasks. Now, I want to finish by mentioning the role of sleep in synaptic homeostasis. This is what we have been testing in our lab over many years. And we have done so because we have started from the very same definition of sleep that I mentioned at the beginning of my talks, which is that what really defines sleep relative to any other behavioral state is a partial sensory motor disconnection, the fact that when we are asleep, our brain is offline. And because this is so dangerous potentially, we think that this means that the function, the essential function of sleep must require the brain to be offline. If this were not the case, evolution would have found a better way, a safer way to carry out the same function without being offline. And we think that function is to maintain synaptic homeostasis. Here I'm showing excitatory glutamatergic synapses because most of the data that I'm going to show you refer to the mammalian brain, in which the majority of synapses are indeed excitatory glutamatergic synapses, usually of this kind, axon-spinous synapses. You see an example here of a electron microscopy imaging. You see the dendrite, the shaft here, and coming out of this a mushroom, a big, large mushroom spine. Here is the axonal button. This is the postsynaptic density. And on the other side, this is smaller, stubby spine. Now, of course, you know that there are many, many synapses. It has been calculated perhaps 100 trillion just in the human neocortex. And as I mentioned at the beginning of my talks, of course, synaptic signaling activity accounts for the bulk of the brain's energy budget, overall perhaps 70% of that. So it is crucial that synaptic activity is kept in balance because the brain cannot afford to spend too much more energy in sustaining synaptic activity. Now, this hypothesis that we have been testing is called the synaptic homeostasis hypothesis, or SHI for brief. And very simply, it states that when we are awake, we are always learning something new. There is incidental ongoing learning, and the end result of this is a net increase in synaptic strength, in the potentiation of many synapses across many circuits in red here. And then the job of sleep would be to renormalize overall synaptic efficacies or synaptic weights, to rebalance, so to produce a net decrease in this strength, so that the next day the overall balance is reestablished. Now, these are the major points of this hypothesis. We must learn during wake, because that's when we are exposed to the environment, we are connected, and therefore we need to learn about the causal structure of the environment. And we know that sleep happens mainly by strengthening synapses, so through synaptic potentiation. This is not controversial. It's also not controversial that because of that, you need rules of renormalization, again, to keep the synaptic weight in balance. So for instance, here are just a few examples of the many studies using computer simulations and neural networks, showing that when you implement rules of learnings through potentiation, as we know they happen in the brain, you also need then rules of renormalization to avoid otherwise the very rapid saturation, increase in saturation in synaptic weight, so runaway potentiation. And of course, once the synapses saturate, there is no longer ability to do new learning. Now, again, this is not controversial, but most people in the field perhaps they still think that the brain is smart enough to be able to implement this renormalization at any given time. So we learn something, we strengthen some synapses, and at the same time, other synapses get weakened so that the balance is maintained. Instead, what we think is that the balance overall is kept within the 24 hours of the sleep-wake cycle, but not at any given moment. So during the day overall, there is an imbalance, so there is a net increase in synaptic strength, again, because I'm exposed to all the images, everything that I see around me. And of course, there are in the brain during wake all the arousal systems, acetylcholine, noradrenaline, histamine, orexin. All those systems are active, and that in itself provides a neuromodulatory milieu that is extremely conducive to synaptic potentiation. On the other hand, instead, when then we go to sleep, that's the preferred time for the synaptic renormalization that we call synaptic down selection, because it should be selective, as I will mention at the end. And it is crucial that we are disconnected, that we are offline, because it's only in this state that we can reactivate during sleep the great majority, if not all, synapses, because neurons are active quite a lot during sleep, and do this comprehensive sampling of all the memory traces, allowing then this smart down selection. The fact that during sleep all those arousal systems are generally speaking less active than during wake is also highly conducive to synaptic depression rather than potentiation. Now, what is the evidence that we have for this major claim, meaning that by the end of wake, synaptic strength overall is higher, and then it gets lower after sleep? It's not easy to do so, to test for this, because there isn't a single way to measure synaptic strength. And so we have been using many measures across different species, literally from flies to humans, under the assumption that if synaptic homeostasis is an essential function of sleep, it should apply to all animal species that have been shown to sleep so far. So we have used, as I mentioned, different methods, electrophysiological, molecular, or ultrastructural measures. For instance, at the electrophysiological level, of course, you can stimulate the neuron A and then record the monosynaptic response from neuron B, which is what is shown here. And of course, the amplitude of this first component, or the slope of this, is a reflection of the strength of the connection between A and B. And these kind of studies have been done both in rodents and in humans. Here you see an example by Huber and colleagues using transcranial magnetic stimulation, so direct stimulation of the cortex in a non-invasive way. And here is the slope of this response, keeping, of course, the stimulus of the same intensity. And you see that the slope progressively increases during the day. It gets even steeper after a night of sleep deprivation, and then it decreases after recovery sleep. Now, similar studies have been done also in rodents, but although this finding is in agreement, is consistent with the major claim of the Schei, there could be other explanations for these results. For instance, overall change in the balance between excitation and inhibition could result in the same finding. More direct evidence comes from the studies of minis, of the miniature excitatory postsynaptic potential. Here is one study that we did in which we measure the frequency and the amplitude of this spontaneous event in the cortex of rats and mice in condition of after we took the brain after several hours of sleep or sleep deprivation or spontaneous wake. And the result was consistent, meaning after sleep overall both the frequency and the amplitude of these events is reduced relative to both the forced wake and spontaneous wake conditions. Now, since then, and I won't go into these details, but there have been at least 3 other studies basically reaching the same evidence that at least in cortex, at least in the superficial layers, overall, the minis seem to suggest this increase in synaptic strength. Now, what about the molecular markers? You can use presynaptic markers, which we have been using in flies because in flies, they are a very good way of tagging and labeling many, many synapses, while in rodents, the best molecular markers of synaptic strength are on the postsynaptic side, and specifically the expression of AMPA receptors. And you see here an example in flies, in which you can see that the expression of this BRP protein that is very widespread in most synapses in the fly brain. The expression of this is increased overall in many fly brain regions, as shown here, after a night of sleep deprivation relative to a night of sleep. The study on the right instead focused on specific Drosophila neurons, specifically visual neurons. They have a very stereotyped morphology. They look quite like mammalian neurons with dendritic branches and spiny dendrites. And what we found was that both the length of these branches and the number of the spines is high if the fly has spent several hours in an enriched environment. After that, if the fly is allowed to sleep, both number of branches and spines go down. But very crucially, instead, if the fly is kept awake for a few more hours, this renormalization in spines and branches does not happen. And this is very important because it's telling us that once you have this kind of growth and increase in synaptic strength, you just cannot renormalize simply because of the passage of time. You need the sleep to do so. Here is the evidence in rodents using the AMPA receptor expression, rather especially the GLUE1 subunit the expression of this subunit and the phosphorylation level of these subunits in specific serine residues, such as the serine 831 or the 845. Those are very well established markers of synaptic strength. They have been known in many, many studies to go up with a synaptic potentiation and go down with synaptic depression. And you see here example in one study that we did in the rat cortex or hippocampal formation. If you take the brain of these animals after several hours of sleep or spontaneous wake, or not shown here, but we got the same results after a few hours of sleep deprivation, and then you extract the synaptic proteins and you run Western blot analysis in synaptoneurosomes. So you are enriching for these synaptic proteins. And what you see here is the overall expression of these markers is 20–30% higher in the wake condition relative to the sleep condition. And more recently, this is a study done by a different group, Rico Ugenir's lab, finding very, very similar results. Now, they focus actually on the mouse forebrain, and specifically they looked at the postsynaptic densities. And the 100% line that you see here for them is the wake level of animals that spent several hours awake at night, spontaneously awake. And relative to that, these are the overall decrease in the expression of several of the markers that I just mentioned after 3–4 hours spent asleep. So these are, I think, we have consistent evidence across labs that when you focus on excitatory synapses in large brain regions, and specifically you enrich for synaptic proteins, you see this quite consistent difference in expression between sleep and wake. Now, more recently, we thought that perhaps one of the stringest way to test this general hypothesis was to measure the size of synapses, because there is a very good, strong positive correlation between functional strength and size. So to do so, we resorted to serial electron microscopy using the block phase serial electron microscopy. You see here our microscope in which you can cut automatically thin sections, in this case of the mouse cortex, and you can overnight obtain large stacks of images that then you reconstruct manually. And so this is actually quite time consuming. So we were interested, of course, in segmenting, reconstructing the dendritic branches, the spines, and then the axons passing by. And what we measure specifically is what you see here in red, which is called the axon-spine interface, which is the direct area of contact between the axonal button in orange here and the spine head in yellow, because we know this is a measure, a structural measure of synaptic strength. Of course, we are counting on the large evidence already present that indeed there are, first of all, very strong correlations between structural and functional measures of synaptic strength. For instance, you see here that either the volume of the spine or the synaptic area, the postsynaptic density, or the axon-spine interface, again, are very strongly positively correlated with the number of the AMPA receptors that are expressed in those synapses, or electrophysiologically the currents that are mediated by these receptors. So indeed, through these studies overall, we find that indeed overall the majority of spines that we tested so far in cortex and hippocampus go down in size after several hours of sleep relative to several hours of wake, either sleep deprivation or spontaneous wake. Here is the original study that we did in the primary motor and sensory cortex in the superficial layers. And what you see here, the single dots are the size of this axon spine interface in single synapses. And you see here that overall, the sleep group on average has smaller sizes relative to the two spontaneous wake and enforced wake groups that instead are indistinguishable from each other. And this shows the same data in this logarithmic scale in which you can appreciate that the overall, the sleep group, the curve, the blue sleep curve shifts to the left, suggesting actually scaling of this size with sleep. This is happening for the majority of synapses. On average, they decrease in size by 18%, but it's actually not global scaling because the largest synapses actually do not seem to scale. So the very, very large mushroom spines do not seem to change between sleep and wake. Now, very similar results, I won't go into the details, were obtained when we looked in the CA1 region, and specifically we focus on the CA3 to CA1 synapses. The findings overall are a little bit more complicated, but the take-home message is the same. Basically, when you compare sleep with spontaneous wake, with sleep, there is a decrease in the size of synapses, especially of these large synapses. And instead, when you compare sleep with extended wake, so with sleep deprivation, is all synapses that decrease with sleep. And actually, they decrease not just in size, but also in number. So what happens during development? Because presumably early on, I mentioned this at the beginning of my talks, of course, there is much more sleep early on in development. If sleep is so important for plasticity, these processes of renormalization should perhaps be present, or even in a more significant way in young animals. And so far, this is indeed what we see. These are 2-week-old mouse pups. And in this experiment, we have two conditions: a few hours of sleep deprivation relative to sleep. We don't have spontaneous wake because these animals never stay spontaneously awake for several hours. But in these two conditions, as you can see, it is obvious that indeed there is a decrease in size of the cortical synapses. This is primary motor cortex. If anything, the decrease is even larger, is actually 34%. And here we see that all synapses actually go down in size. So it seems to be a quite pronounced scaling. Now, this is for the evidence. Now, the interesting question remains, can we, through this process of down selection, account for some of the beneficial effects of sleep that I've been discussing? So the effects, of course, are at the cellular level as well as at the systems level. At the cellular level, perhaps it's not too difficult to imagine that indeed, if you, by the end of sleep, you overall decrease the size and the strength of many synapses, you end up with a brain that is leaner and requiring less energy. And so there is a benefit in terms of, if you wish, energy restoration. But I want to pinpoint that this is not the same kind of restoration as described at the beginning of my talk, when you think it is not in line simply with the general idea that, well, with sleep, we save energy simply because neurons are less active, they are firing a little bit less. here the restoration and the saving comes simply because the sleep is thought to every day allow the brain to go back to a sustainable level of energy. So it is in the long run that this saving would make sense, again, to avoid that you build up synaptic strength to a point that becomes totally unsustainable from an energy standpoint. Now, the other point, of course, is that if you bring down overall synaptic strength, you avoid synaptic saturation. So you move away many synapses from the level in which new learning, new potentiation would not be possible. And we have some evidence for this. For instance, in animals, you can test the ability of the brain to undergo further synaptic potentiation by using optimized paradigms of long-term potentiation in the brain of animals that have been awake several hours or asleep several hours. So here is one of these experiments. So here in vivo, LTP was induced electrically in the cortex. It was always induced in the same condition of quiet wake. The only difference between the two groups was that in the wake group, animals spent the prior 3-4 hours awake, while the sleep group had been allowed to sleep. And so here 1 is the starting point, and 2 is the amount of potentiation that we were able to get applying this LTP protocol. And you see that there is very little increase if the animal had been already awake for several hours. Instead, there is a significant increase after sleep. So this would suggest that, in fact, just being awake for a few hours does bring the brain closer to the level of saturation. And similar results have been obtained actually by this group of German researchers in humans using TMS to try to induce an LTP-like plasticity. And they use the same paradigm. And again, here they had 2 groups of subjects that before the paradigm was applied had been either allowed to sleep or sleep deprived. And you see that the same, there is, each dot here is a subject, so there is very large inter-individual variability, but overall, it's obvious that the same kind of stimulation produces, in most cases, LTP-like increase in the response in the subjects that could sleep before the experiment. And instead, this is not the case in the sleep-deprived subjects, in which actually there is, if anything, an LTD-like, long-term depression-like response. So, of course, the moving away synapses from saturation may very well be the reason why, as mentioned before, we know that sleep promotes future learning. So perhaps the most difficult still and open question in our field is how can we reconcile this overall effect of sleep for synaptic renormalization with memory consolidation? Because you could imagine that many people equate memory consolidation with synaptic strengthening. And so it's an open question in the field whether indeed this memory consolidation happens because in sleep some traces get further potentiation, or they simply get protected and they don't get depressed relative to the others. This is something that we don't understand. And we don't simply because it's very difficult to perform experiments that can allow us to really fill this gap between the synaptic level and the behavioral level. So here is an experiment that we have recently done to try to address this specific question using a complex wheel task in which the mouse is learning how to stay and run at progressively higher speed on this complex wheel whose rank pattern is uneven. It's 1 hour of intense training that is applied once a day. And we have consistently found that indeed the mice are very able to learn this task in one session of 20 trials. So they're able to get faster and faster before they fall out of the complex wheel. Now, the result is the following. After they learn, they either are allowed to sleep for several hours or they're sleep deprived. Then the next day, they are tested, and we see consistently that the sleep group starts from the same level as at the end of the session 1, and instead the sleep-deprived group has degraded performance. So we can definitely say that this, in this task, sleep promotes offline consolidation. Now, in a recent study, we ask, can we see what happens to the specific synapses in the primary motor cortex that we know is the area involved in learning this task? What happens to them before and after training and before and after sleep deprivation? So we use this paradigm in which we have mice, in which we can really visualize the dendritic trees of the pyramidal neurons in motor cortex, and specifically then measure in the spines one marker of this GLUE1 expression. In this 2-photon repeated imaging experiment, we can image the same spines, the same dendrites, before and after sleeping baseline, then after training in the complex wheel task, followed by then either sleep or sleep deprivation. And then we also, we can do training, testing, and imaging the next day. So the point again is to be able to follow the performance, follow sleep and sleep deprivation, and at the same time look at each single synapse to see whether they potentiate or not. And so here is for all the animals that we have, 12 mice, we were able to image across 5 different sessions, more than 1,500 spines. And what is shown here is the overall change, the normalized difference in the expression of this marker of synaptic strength for the entire population of spines. And so perhaps not surprisingly, because this is very consistent with what I've shown you before, in baseline condition, at the end of 6-7 hours of sleep, the overall expression of this marker goes down. Then also not surprisingly, immediately after training, the expression of this marker is increased. Because again, we have known before that this training leads to synaptic potentiation in the motor cortex. Now, what was new is the following finding, meaning that even immediately after training, if the animal is allowed to sleep, the overall net result of sleep is a decrease in synaptic strength. Instead, if the animal is kept awake, this does not happen. So this is the first result, but of course the most important for us was what happens specifically to the synapses that are engaged in learning. Because not, of course, all synapses in the primary motor cortex get potentiated because of this learning. We found indeed that 10% or so show a large increase immediately after training relative to before, while 90% or so did not. So we call these the max spines and the other spines. And this is very consistent with other results from other labs. So then we ask for these max spines, how do they behave in sleep and sleep deprivation relative to the other spines? And this is the result, basically. So here I'm showing the two groups, the sleep and the sleep-deprived group. Of course, up to this point, the two groups are experiencing exactly the same conditions. And by definition, these are the max spines. So we select them as the ones that show the largest increase relative to before training. What was intriguing and surprising to us was the fact that after the session of training, these spines behave exactly in the same way in the sleep and the sleep-deprived groups. So they stay relatively high. They are still, even 24 hours later, more potentiated than before training. But there is no difference between sleep and sleep deprivation. The difference comes from the majority of the spines, the other spines that indeed get depressed, depotentiated. They renormalize with sleep, but of course they keep the same level, the same strength with sleep deprivation. And so at the end, you can say that sleep affords a relative protection to the max spine, simply because although the max spines per se are affected equally by sleep and sleep deprivation, at the end of sleep, the max spines are in a better relative position because all the other spines got depressed. And this is what basically is shown in this graph, showing that again, if you express the difference in the strength of the max spines relative to the other spines, then the max spines are in a better position after sleep than after sleep deprivation. We also found that the ability of the animals to perform better the next day, to show this offline consolidation, depended on how much the majority of spines, the other spines, got depressed after sleep. So again, it seems that the weakening of the other spines is what accounts for the better performance the next day. So if we put all this together, therefore, at least in this specific task, it seems that sleep helps because it increases the signal to noise ratio, but it does so not because it increases the signal, the max spine's strength, but because it decreases the noise in the other spines. And by doing so, it seems that sleep can get many benefits altogether. Because of course, by depressing many spines, you get energy saving. You move again away spines and synapses from saturation, so you promote further learning. And at the same time, by this relative protection of the max spine, you consolidate the memory in this task. So what are the mechanisms of this synaptic down selection? This is what is being investigated in our field by many different labs right now. We know that the fact that the neurons are active in a very specific way through the slow waves and the up and down state in slow wave sleep, the ripple activity in the hippocampus, those specific patterns are important and are actually required for down selection. We have also identified in the field some of the major molecular players that may be critical for the weakening of synapses during sleep. And perhaps the best established right now is the HOMER-1A, as demonstrated by Deering and colleagues. I thank you very much for your attention.